Okay, welcome to the Karst Waters Institute Frontiers in Karst Colloquium, where we're starting off today with our first talk for the spring series. And we're continuing in a series of talks related to critical zone processes in carbonates. We're still working on organizing the next couple of talks, but there will be two more talks, one in March and one in April. And you should expect to see some dates and speaker names on those probably in the next few days. So today we're excited to invite Dr. Liz Hassenmueller from St. Louis University to speak to us today. And she got her undergrad at Indiana University. She was a double major in geological sciences and biology, which I think will feed well into the research that we'll see today. Then she went on to get a master's and PhD at Washington University in St. Louis, followed by a postdoc at Washington University, and then a postdoc at Penn State, working, I think, with the Shale Hills Observatory there, Critical Zone Observatory. And today she's going to be talking about how roots impact weathering. So with that, we'll welcome Liz and hear what she has to say. So I'll stop my sharing here and you should be able to take over.
Okay, perfect. I'm gonna shut my video off just to help with bandwidth. So I'm going to click a few settings here and share my screen, which is working. Okay, great. Yeah, so thanks for that introduction, Matt. As Matt mentioned, I'm Liz Hassenmueller and I'm at St. Louis University. And the focus of my talk today is going to be on weathering of rock to regolith, the activity of deep roots in bedrock fractures. And as Matt mentioned, this is work that's focused on the Shale Hills Critical Zone Observatory. So obviously very different lithology than probably some of your foci, but I want to relate some of the work that I did there in a silicate terrain to carbonate terrains throughout the talk. So the focus is going to be on this shale lithology, but I want to intersperse some ideas related to carbonate terrains as well. So to get us started, I did want to acknowledge my collaborators on this project, including Jin Gu, Julie Weitzman, Tom Adams, Gary Stinchcomb, Dave Eisenstadt, Patrick Drohan, Sue Brantley, and Jason Kay, as well as the funding for this project from the National Science Foundation. So I also wanted to give you guys just a brief introduction to some of the work that I do in carbonate terrains. And so this is one of my field sites where I've been doing work for the past 5 or 6 years, which is a carbonate spring system here in Missouri. So I live in Missouri. We have wonderful carbonate terrain. So many of the projects that I'm working on are focused on carbonate systems. And to get us started, I thought one of the most relevant research areas right now, as half the country is coming out of this deep freeze, is the work that my group has been doing looking at road salt applications. So road salts used to de-ice roadways, as you can see here in this image taken by one of my graduate students, and how that material is transmitted through soil environments into the subsurface, into these carbonate aquifers, and how that material can be transported rapidly through conduits or more slowly through the rock matrix. So my group's been working on this for several years, and I've listed a few of those publications below, as you can see here. I've also looked at tracing water flow paths and carbonate systems, looking at how carbonate aquifers interact with surface water systems, as well as how water flows through carbonate aquifers to spring systems as well. And then lastly, some of my more recent work has been focused on emerging contaminants like microplastics and how they're sourced to carbonate aquifers, as well as how they're transmitted through those aquifer systems. So this is some work that one of my graduate students is currently doing. Looking at microplastics, you can see an assortment of microfibers that have been collected from water samples in a cave system that we've been monitoring for about the last year. So these are some of the projects I've been actively working on in carbonate terrains. But as I mentioned, my focus today is going to be on a silicate system where we have shale lithologies. And I'm specifically going to be talking about the microenvironments in rock fractures. which are really understudied due to the difficulties of accessing these environments. So really there's not much that's been done with these microhabitats just because of the difficulties in getting to that deeper rock layer and extracting the rock and looking at these systems. So plants, we know, play a key role in weathering rock to soil in the critical zone, and plants aid in mineral weathering reactions through several processes, including the production of CO2 through root respiration, as well as the production of organic acids like root exudates that enter the soil system or other systems and can cause mineral weathering reactions. And these mineral weathering reactions are quite important when we think about controls on rock porosity as well as permeability, the global carbon cycle, as well as climate change. The role that these plants play in weathering rock varies as a function of many things. Some of the important components are the water availability and use of the plant, the associated mycorrhizal fungi community around the root system, and then the root depth and distribution. And so the focus of my talk is going to be on this idea of rooting depth and distribution. And here we have that classic figure from looking at the critical zone. So we have a slice out of the critical zone where we can see trees and their roots, and those roots are relatively shallow in most cases in these weathering systems. Here we see another example from Brantley 2010 showing these shallow rooting systems. So when we think about the weathering potential of tree roots and how it relates to the depth and distribution, we know that most roots are commonly located in the top 30 centimeters of soil. So over 50% of roots globally are in this top 30 centimeters of soil, and 95% of all roots globally are located in the top 2 meters of the subsurface. So we generally assume that rooting depth is really restricted by underlying bedrock. However, many landscapes are characterized by soils that are underlain by actively weathering bedrock. So these roots may be restricted, but deep roots can be quite important in these systems, as we see when we look at the entire picture from Brantley 2010. We can see that many of the roots are isolated up in the upper portions of the soil, but we also have these deeper roots which can penetrate into rock fractures, and so we can have the potential of soils, gases, water, and roots moving deeper into these rock fracture environments. So the weathering of the potential of tree roots depends on, as I mentioned, climate, particularly precipitation and evapotranspiration, the tree species themselves, as well as soil thickness and soil properties, but also these bedrock properties. And I know that this group is focused on carbonate systems, so I wanted to contrast differences between different types of critical zones. And so here I've got the Carbonate Critical Zone Research Coordination Network's really nice figure from their website showing the differences between weathering zones in silicate critical zone systems shown here on the left side of the screen and carbonate systems shown here on the right side of the screen. And you can see immediately that they're fairly different carbonate or critical zone systems because of the substrate that you're dealing with. So weathering zones in silicate systems tend to be much shallower, from 10 to 100 meters, whereas in carbonate systems, we can have much deeper weathering zones from 10 to 1,000 meters into the subsurface. So then when we look at these bedrock properties and compare them, looking at Wendy Graham's figure from a 2012 paper, we can see that in silicate critical zone systems here, some roots have been plotted. We can see the weathering zone, and we know that in these systems, permeability tends to decrease rather consistently with depth in silicate systems. If we contrast that with the carbonate systems, which again have these much deeper weathering zones, we know that we've got much more heterogeneous permeability, and that heterogeneous permeability that we see in the carbonate critical zone weathering system enhances the connectivity between surface systems and the subsurface. So this has some important implications for how roots might be behaving in silicate critical zone systems as compared to carbonate critical zones. So the vast majority of studies that have looked at deeply rooting systems have really focused on the water holding capacity of weathered rocks, either in arid environments or drought prone environments where you may have a dry season that decreases the amount of plant available water. So here I'm showing an example of a study that was done in the Yucatan where they were exploring this idea of rooting depth, but focused on how that relates to water stress in the system. So they had a deciduous tree system, a soil that overlaid these carbonate rock types that are shown here in this profile. And what they did is they looked at the water content in different potential reservoirs that trees might be accessing in this system. So on the right side of your screen, what you're seeing is the volumetric water content for different reservoirs, including shallow reservoirs like soils compared to deeper reservoirs like the rocks, the underlying carbonate rocks below. The blue bars in both of these plots are indicating rainfall amounts, and then the lines that you see with points in between are showing either soils or the volumetric water content in rock. What they found was that while the soil tended to have higher volumetric water content, the vast majority of the total volume of water being stored in the system was below the topsoil, and dissolution features in these carbonate rocks allowed these deciduous tree species to allow their roots to penetrate deeper into the weathering profile and actually access the underlying aquifer below. And so the hypothesis was that these tree species were penetrating deeper into this weathering profile in order to access these deeper water reserves whenever there might be times of water stress. Similarly, there have been a lot more studies that have been conducted looking at deep rooting systems in granitic terrains, and specifically in drought-prone or arid granitic terrains. So these are some figures from Robert Graham's study in 2010, showing plant available water as a function of the growing season in different reservoirs. So the soil shown here in gray, and then deeper water reservoirs in the sap rock shown in the black down here. And in both cases, water levels decrease over time, but the soil water ends up having much lower water volumes, and that eventually kind of peters out midway into the growing season, showing that these plants are in fact experiencing water stress. And their study showed that these plants are sending their roots deeper in between joint fractures in these granitic rock terrains so that they can access deeper water. The other figures that you're seeing in this diagram are showing basically a dye trace that shows the water flow pathways in the field, and then also at smaller scales between fractures in the rock. So then the question becomes, what do tree species do in more humid environments where there's not the same kind of water stress that you see in arid or drought-prone environments? And so studies that have been conducted in more humid environments, specifically in central Pennsylvania, looking at the Shale Hills catchment, have shown that tree species tend to rely on these shallow soil water reserves rather than deeper water reserves as seen in the more drought-prone environment. So this is a plot showing water isotopic composition, so deuterium plotted here on the y-axis and oxygen 18 on the x-axis here, for various types of water in reservoirs found in the Shale Hills catchment. So those reservoirs include things like precipitation, which are shown in diamonds, as well as deeper soil water reservoirs shown with the circles in gray shading here, groundwater over here, that all plot along the meteoric water line. Over here in the circles that have various colors, these are isotopic compositions in stem water for various tree species. And you'll notice that those tree species tend to have very similar isotopic composition to the soil water that's relatively shallow. So this study by Gaines et al. In 2015 concluded that these tree species are really reliant for the vast majority of their water on the upper soil horizons. So it becomes a little bit unclear in these humid environments why, if there are deep roots, and if so, why are they there and what are their microhabitats? So the focus of my talk is to understand these processes using the Shale Hills Experimental Watershed as a case study for root rock interaction. So here we can see Shale Hills in central Pennsylvania on the old NSF map of the former CZOs. And here we see where Shale Hills is in Pennsylvania in the center of the state. And this is the well-studied Shale Hills catchment over here. We focused our study on the Misgrouse Gulch catchment, which is nearby, but it's about a quarter of a kilometer away. It's just one catchment north. So we wanted to look at what were the depths and distributions of tree roots in these more humid environments, and what characterizes that deep rooting environment. So what is the microhabitat? If there are deep tree roots in rock fractures, what's that microhabitat like? And then what is the potential for these tree roots to weather bedrock? So these were the three core questions for this study. As I mentioned, the focus of our study was the Misgrouse Gulch catchment, which is very close to the well-studied Shale Hills. We were unable to do our experiment in Shale Hills because of the threat to ongoing experiments, because we were excavating the soil and into the rock. And so we didn't want to disturb anybody else's experiments. So we moved just slightly north where we had very similar types of tree species, the same shale bedrock, the Rose Hills Shale, and very similar types of soils. So we excavated pits along a ridge top, mid slope, toe slope, and valley floor on this north hill slope in the Misgrouse Gulch catchment. The way that we excavated those pits was to first use a track hoe to remove the topsoil, and then we dug all the way down to where we could no longer use the track hoe to remove material. So this got down usually to sort of weathered rock at this point. Once we were no longer able to excavate with a track hoe, we used an electric jackhammer to get deeper into the rock profile. So you can obviously see that these kinds of studies tend to be limited just because it's very difficult to dig into the rock to explore these microhabitats for roots. So once we had excavated the pits, the next step that we took was to look at root respiration. So we extracted roots from the pits in the soil as well as below the soil in the rock fractures, and we measured the root respiration rate in the field as shown here in these top two photographs. We also collected soil CO2 from the soil atmosphere as shown here with these soil CO2 samplers. And we did that before, during, and after the experiment to assess the variability in soil CO2 concentrations as a function of time. Once we completed these two steps, so we extracted the roots, we then mapped and imaged the roots and rock fractures on the pit surfaces. Obviously, we weren't imaging right where we had extracted roots, but we imaged where we had not disturbed the pit surfaces. And so we did that for our ridgetop site, mid-slope, toe slope, and valley floor site, as you're seeing here in these top pictures. This is what each one of the pits looked like. The yellow line is denoting the difference between the augerable regolith up here at the surface. So this is what we could auger through by hand. And then below that is material that we could not auger. I'm also showing an example of a fracture along this red line with the asterisk. So you can see that fracture here, but there were many fractures going across the surface of the shale in all of the different locations that we sampled, except for the valley floor, which you can see there's no yellow line delineating the difference between the augerable regolith and rock, because we weren't able to reach rock at this particular site due to the depth of the soil. So we went about 2.5 meters into the soil and were unable to reach the rock. We were kind of constricted by the pit stability as well as the length of the track hoe arm. So we were unable to reach the rock at this location. So here are some examples of some of the roots and fractures that we imaged on the surface of the pits. And so all of these roots and the rock fractures that you're seeing are at about the same depth, 1.2 to 1.3 meters below the surface. And you can see those microhabitats where we were finding the roots, as well as this fill material that you can see in the field image, but a little bit better here in an SEM image that shows roots embedded in this rock fracture fill material. After we had completed all of our imaging and mapping of the pit surfaces, we then collected physical samples of soil, rock fracture fill, and analyzed those for various physiochemical properties, including the elemental composition, the mineralogy, the morphology, using thin section. So you can see examples of us collecting some of these samples, as well as some of the samples that were extracted from the pits. So now to orient you, I'm going to be showing you a series of figures that look similar to this, where we have each one of our pits from the ridge top all the way down to the valley floor. And I'm showing the soil horizonation here. Again, this yellow dashed line is indicating the difference between the augurable regolith and material that we could not hand auger, including the rock shown in gray in each one of these figures. And then along here is a series of other pieces of information that I'll cover throughout the rest of the talk. So the first piece of information that I wanted to show here is the fracture volume in each one of our pit sites. So the shapes here are indicating the circles are indicating various fracture sizes going from 250 microns to greater than 1000 microns. So with increasing aperture size, you have darker shading here. And then the triangles are showing the total fracture volume for each one of these sites. And as you can see, that fracture volume tends to decrease with depth in each one of the locations where we were able to reach rock. So we saw this decreasing trend in fracture volume, which is not surprising for a silicate system where we consistently see decreasing permeability as a function of depth. We found the highest fracture volume in our toe slope site, so our downslope site, and we didn't see significant differences between our upslope sites at the ridge top and mid slope. Next, we looked at the roots that were present in these rock fractures, and we found that roots were present in rock fractures even in our deepest, least weathered shale. So this went all the way down to about 2 meters into the subsurface. So you can see roots plotted in a couple of different ways. So the first one is root length density, which is shown here in column C. And what this is the root lengths that we found in various soil depths plotted here as a function of depth in our pits. And these were taken or collected with a soil core. So essentially we cored all the way down to the rock to get the root length density. We were unable to collect similar cores for deeper samples because of the difficulty in excavating the rock. So the way that we looked at root densities deeper in the profile was from our photographic imaging. So here you're seeing intersections as a function of area in the pit, and you can see that those root densities decrease as a function of depth in all of the places where we sampled. And what we found, interestingly, is that root densities were lowest in the toe slope. So here you can see in where we got to the shale in the toe slope that the root densities were much lower, despite the fact that we had higher fracture density. So while there was more area for these roots to go, we saw fewer of them, and we suspect that that's because they were restricted by the water table at this location. So we didn't see, it was fairly dry when we sampled our pits, but when we came back, we could see that the water table had come closer to the surface and that we had water in the pits. We also found water in our CO2 samplers, indicating that this water table is very shallow at the toe slope. When we started looking at the material in these fractures, we found that roots were always associated with this fill material that you can see shown in this close-up picture in the field. So we have a root coming through here, and then you can see this particulate material surrounding the root. And anytime we found roots, we always found this fracture fill material. We also only observed roots in fractures that had apertures wider than 50 microns. And to show a little bit closer up, here's a thin section where we have a cross section through a root shown here. And you can see this particulate material that surrounded this root, and that there's this opening in which the root is growing. So we always found that roots were in these wider fractures, and that the fractures were always filled with particulate material. So So in terms of that fracture fill material, that material was similar in terms of its clay composition and elemental mobility to the lowest soil horizon. So the chemistry of this material was very similar to overlying soil horizons. And I want to focus a little bit on this idea of elemental mobility in the next couple of slides. So the way that we characterized elemental mobility was using the mass transfer coefficient tau, and we wanted to quantify the chemical weathering in the rock fractures using these values. So for those of you who may not have used tau before, the way that you calculate tau values is that you use the chemistry of different materials. So you have the chemistry of weathered rock, or whatever your sample is, compared to the parent, which is P, and you have some mobile element J that you're interested in. In this case, those are potassium and magnesium, as shown here in the plot. And you compare that with some immobile element I, and I'll talk a little bit more about the immobile elements we were using in just a second. You subtract 1 from that, and the values tell you whether or not you have depletion or addition of your mobile elements. So for example, here I'm showing data from the ridge top, toe slope, mid slope and toe slope, and you can see various values plotted as a function of depth. For tau, we can see some of these kind of hitting around minus 0.5. This is telling us that there's been 50% depletion for potassium in this for these particular samples that I'm highlighting here with my pointer. If we see a positive number, then there's been some sort of addition relative to the parent material. So for our tau assessments, we selected 2 immobile elements, the first being zirconium, which is shown here. The data are shown as triangles in these plots. And this choice of immobile element tells us about elemental losses as solutes as well as particles. So in our system, zirconium is largely in zircons, which are relatively insoluble and also relatively immobile compared to other types of particulates because they're rather dense. We also looked at tau using the immobile element aluminum, and so that's shown as these circles in all of the plots. And so aluminum tells us about the elemental loss strictly as solutes, because in our system, aluminum is relatively insoluble, but it can be more readily transmitted as particulates because it's associated with clays, which are less dense and a little bit easier to move than zircons. So looking at our data, again, the yellow line indicates the difference between the augurable regolith up at the surface and then more material that wasn't able to be hand augured. We found that in our soil that we had depletion in all of our samples shown in the blue here. So we saw these depletion effects in the soil, and that reflects the weathering of illite and vermiculitized chlorite to kaolinite in our system. If we go deeper into our pits and look at the shale, what we found is that it was similar to deep unweathered parent materials. So you can see that tau values are very close to 0, indicating that there's not a lot of elemental loss for either potassium or for magnesium in these cases. When we look at the rock fracture fill, which is shown with the red symbols here, what we found is that it's relatively similar to the soil. So the fracture fill material had very similar tau values. So that begs the question, did we have some movement of soil downward in our system? Well, our data indicate that this rock fracture fill material is actually not translocated downward. It's instead formed in situ through chemical weathering processes. So the idea here is that clay minerals are easier to translocate than zircons from the surface, the soil surface, into these rock fractures. So if we use zirconium as our immobile element, those tau values should be higher for fracture fill. So here's our actual data, and here's a predicted value if that material is translocated from the surface. So the gray point is showing what we estimate if we were moving soil into our fracture. However, we don't see that. We see that the fracture fill is very similar to the soil, and that indicates that that material is the result of pedogenic processes rather than translocation of that material downward. So I also wanted to note here that when we were looking at this material in the field, we also saw that we had fracture fill lining horizontal fractures, not just vertical fractures. So this would also be indicative that that material was forming in situ rather than being translocated downwards. So next we wanted to examine the microhabitat for roots in these bedrock fractures. And what we found is that we had accumulation of CO2 at depth, and that could potentially provide a source of acidity for bedrock weathering. So the plots you're seeing, A through D, we've shown before. E and F are showing some of the CO2 data that we collected. So the column E is showing the root respiration rate. Each one of these dots is representing an individual root sample that's been normalized to the root's mass. And so what we found is that the root respiration rate really didn't change all that much with depth. So we didn't see that the roots were necessarily behaving differently as a function of depth. However, the total CO2 flux from root respiration tracked with root density. So you can see the root densities are going down. And so the total root respiration decreased with depth because you simply had fewer roots deeper in the weathering profile. However, what we saw when we looked at our CO2 data is that CO2 in the soil accumulated as a function of depth. And that's largely because of the slow diffusion of CO2 out of the profile. So we had the accumulation of CO2 with depth, which could provide this source of acidity along with potentially root exudates that might be introduced into this microhabitat. So then the question that I posed before is what's the reason for deep roots in these fractures if we don't think that they're trying to get down there to access water? So we looked at several micronutrients in the fracture fill and compared that with the lowest soil horizons. We also looked at the plant available water and did some comparisons there as well. And so you can see again our different pit sampling sites shown here and the volumetric water content for those different sampling sites. I mean, the gravimetric water content for those different sampling sites. And you can see that that water content generally decreased with depth in each one of our pits that we sampled, where we got to about 75% less water in the rock fractures as compared to the overlying soil. So while these are point measurements, the fractures were not consistently wetter than the soil above. We also saw that bulk phosphorus was lower in our fractures compared to the overlying soil horizons, and we had similar extractable dissolved organic carbon and bulk calcium in the soil compared to the fracture fill. Interestingly, we saw that we had higher inorganic nitrogen species, including nitrate and ammonium. So those could be anywhere from 4 to 7 times higher in the fractures compared to the overlying soil horizons. And when we took a look at things like bulk potassium, what we found is that while the fractures so the fractures are being shown here in red, the fractures had lower overall bulk potassium compared to the rock samples, we did see that the concentrations of potassium in the rock fractures tended to be higher than what we observed in the overlying soil horizons. So while I'll make a couple of notes here that we did measure bulk concentrations of things like potassium, calcium, and phosphorus. And so these don't necessarily represent the most bioavailable forms of these nutrients. So future work would really benefit to look at the bioavailable forms of these different nutrients in these fracture habitats so that we could get a better assessment of what is actually available for plant roots deeper in the weathering profile. The other thing I want to note too is that while some of these species are higher in the fractures, because there's a limited number of roots deeper in this weathering profile, the bulk majority of what the plants are accessing would still be coming from the overlying soil horizons. So then if we look at root rock interactions in these silicate critical zone systems, in terms of depth and distribution, roots and the fill that was found in these rock fractures, we observed those all the way down to about 2 meters. And those materials, both the roots and the material in the fracture, were very similar to the overlying soil horizons, though they were simply less abundant. So we were basically seeing the same kinds of things in the fractures, but just less of it. In terms of the deep rooting environment, the roots may take advantage of higher nutrient availability in these rock fractures, but it would be beneficial to do additional work to think about the bioavailable forms of the different nutrients in rock fracture microhabitats. We also saw evidence that these deep roots, at least in humid climates, may actually be restricted by the water table, and that inhibits them from sending roots deeper into the weathering profile, which is in contrast to what has been observed in more arid environments where there have been studies of water limitations and how trees kind of handle those water limitations. Lastly, when we think about the bedrock weathering potential, root respiration likely contributes to the in situ weathering that we observed in these rock fractures. So our tau values are telling us that we had in situ weathering processes, and the roots potentially play an important role by enhancing the acidity of these microenvironments. So lastly, I wanted to contrast this, what we observed in the shale system or silicate system, with how these observations might transfer to carbonate critical zone systems. So in carbonates, like I mentioned before, we have higher connectivity between the surface and the subsurface, and there's often been examples of people observing tree roots that penetrate into cave systems. So obviously tree roots are getting into these systems. The permeability is also much more heterogeneous, and the weathering zones are much deeper. And so there's been several studies that have been trying to address the microhabitat of roots in fractures and carbonate systems, including at the Kanza Prairie and the Savoy Experimental Forest, looking at the impacts of CO2, but also in root sway and swelling of roots when they're absorbing water. So there is focus in these carbonate systems as well. So with that, I thank you, and I'll open it up for any questions.
All right, thank you, Liz. So I'll start out with a question. If other people have questions, I suggest you type it into the chat, and Rachel Bosch and I will be reading those out for discussion. So let's see. I had lots of questions, but I'll start with one which relates to some of your data. So when you were looking at the CO2 concentrations with depth, I was surprised how linear those relationships were. Like they really looked like a straight line, which is what you would expect if you had a source at depth and you let it equilibrate via diffusion. So have you guys looked much at those profiles and trying to understand why they have the shape that they do?
So there has been work looking at the profiles. I have another paper that specifically was looking at these behaviors as a function of depth in different slope shapes. So we looked at these planar hill slopes as well as swales, where we have kind of funneling of water into a particular location. And we did see variations depending on water content in the soil profile that weren't exactly linear. So on the planar hill slopes, we did see that linear relationship, but on the swales, we saw behavior that was driven in large part by the water content in the system.
So by the water influencing the transport, you think, or influencing the production?
Probably both. So we did some, when we looked at those data, essentially there's kind of a sweet spot where if you have enough water, you get higher production. If you have too much water, then you get lower production. And then you also have water inhibiting the diffusion. We largely think that what we were seeing was diffusion inhibition in terms of where the water was located in those profiles.
Okay. I don't see any questions in the chat yet. So, oh, here's one from John Martin. Okay, he says, my question is similar to Matt's. I'm wondering if the soil CO2 profiles change with time that would indicate changes related to changing atmospheric pressure. I might add, or temperature to that.
Yes, they do. So I didn't show the figure, but we also were looking at the CO2 concentrations as a function of time. And what we saw is that as temperature decreases, the CO2 concentrations decrease with that. It also is related to the precipitation, so recent precipitation events. You can see variations in CO2 based on how much water is in the system, either enhancing root respiration rates or inhibiting diffusion out of the system. So in the paper that is where this talk is from, there is in the supplemental data to show how the CO2 profiles were varying as a function of time.
Okay, this looks like a follow-up question. John is asking if the change with temperature and water content is caused by production or exchange.
So we didn't specifically look at those processes. So I know that there's some new studies that are coming out with that, but we didn't specifically look at that.
Okay.
We do I was just going to say we did some microbial experiments as well, looking at production rates for samples that we collected from the pits. Those were relatively low compared to root respiration rates. But yeah, we didn't look at that relationship in detail between the CO2 concentrations in the soil versus what we were seeing in terms of our lab experiment.
Okay, thanks. I had a methods detail question, and maybe I missed this, but can you please describe the process for measuring root respiration?
Yeah, so we used a instrument, I'm blanking on the name now, it's Hitchkins, I can't remember now. But basically it measures how much oxygen is consumed, and we assumed a one-to-one relationship between oxygen consumption in that instrument and CO2 production. So actually what we're looking at there from the instrument itself is oxygen consumption, but we assumed one-to-one relationship to derive CO2.
Okay, thanks.
I actually had a question related to that. So when you're making those measurements, are those in situ, or were you taking the roots out and then measuring it like after it's out of the soil?
So it's out of the soil. So what we would do is we would extract the root from the pit surface. And so that was either in the soil or in the fracture itself. We would then clean the root of any debris and we put it into a buffer and then measure that with the instrumentation that I showed in one of the slides. So there is the potential that these roots are, well, I mean, they're clearly stressed when you extract them. So that is something to note in those data, that it was data from extracted roots.
So
go ahead, Matt.
Yeah, no, I was gonna have another question, which maybe also gets into thinking about carbonate. So I'm curious the variety of processes by which roots are getting into the fractures or possibly creating the fractures. Like, are roots able to widen these fractures or initiate the fractures? And in carbonates, are they able to even perhaps dissolve their way into the rock chemically?
Yeah, really interesting questions. So we don't know for certain, but what we were seeing in our data is that whenever there were fractures that didn't have any of this fill material, we never saw any roots in them. So probably the fractures themselves are from periglacial processes that were happening in this system, creating these fracture spaces, and then roots are taking advantage of that opening. And then likely a combination of abiotic and biotic processes are producing these in situ weathering products, CO2 enhancing the acidity as well as root exudates. So in terms of carbonate systems, I think there's these interesting questions you're asking of how the roots get into the fractures. Are they creating them? Are they, weathering? Are they dissolving the carbonate themselves? So I think those are really interesting potential questions to ask in carbonate systems.
I've certainly seen pieces of limestone with just like the print from the roots just dissolved into making little tunnels. So I would guess that there's some of that going on. But
Yeah,
kind
certainly.
of related to that, do you think the fact that we can access the roots in caves, does that possibly give us some interesting things that we can do in the karst environment that we might not be able to do otherwise? Like, can we somehow take advantage of that?
Definitely. I think that there's things that could be done in situ in caves that could tell us a lot about these rooting microhabitats that you can't do in silicate systems. As I mentioned at the start of the talk, it's really challenging to access these things because you're either digging a giant pit like we did, which was not a trivial process, or, you know, some studies have accessed quarries where they can get these rock faces where they can look at the processes, but then you're usually coming in sometime after the quarrying has happened, so the roots are stressed or the environment has changed in potentially multiple ways. So the fact that in carbonate systems you can access some of these roots in the cave itself is a real opportunity to then look at that environment and measure things in situ without disturbing the system as much as you would in the example that I gave today, but also in some of these systems where they've looked at active quarries and things like that. So caves are a great window of opportunity here.
Yeah, I was just thinking about that you've mentioned several times the level of root stress from the research. And I would imagine that, you know, in addition to disrupting them to take samples, you know, just the act of digging the pit is extremely stressful for these plants. And I'm wondering if you can estimate what level of impact that has on the data in this work.
So I think it has fairly minimal impacts on the microhabitat in terms of the bulk chemistry. I don't think there's going to be really anything that's that modified in terms of bulk chemistry. For the extractable chemistry that we did, I think that's probably minimally impacted as well. But the roots themselves are probably fairly stressed. I don't know exactly what that means in terms of CO2 production rates, but probably what we have is a lower end estimate of what they're doing in the experiment that we were doing. So probably CO2 production rates are a bit higher than what we observed in the field.
Okay, we've got another question from the chat from Allison Toon. She asks, did you find many dead roots that were decomposing, or were they primary live roots? Specifically thinking about deeper in the profiles.
Specifically, so we did find dead roots, and in the thin sections that we looked at, we did find evidence of organic material from roots that we saw in thin section. Some of the roots that were in there, there weren't as many dead roots. I'd say maybe 10 to 15% of what we were finding were dead roots, but for the most part, they were living roots in the deeper and shallower portions of the profiles we were looking at.
Okay, I'll ask at least one more question. I think I could sit and ask questions for another half an hour, but I'll save everyone that. But I have at least one more, which again relates some to thinking about carbonates. So you talked at the beginning about how in humid versus arid environments, the water or the trees are getting the roots from, or getting the water from different depths, either from the soil or from the rock. And I'm curious whether that impacts the depth distribution of roots and whether as a result it might impact the depth distribution of CO2 production and weathering, which might in carbonate settings be really an important driver of your patterns of weathering.
Mm-hmm. Can you repeat the first part of that question again?
So
Sorry.
thinking about water availability and how that impacts the depth distribution of roots and or the depth distribution of CO2 production,
Mm-hmm.
which should be really important in carbonates. Like, where is that
Right.
CO2 being produced?
Right. So in terms of the depth of and the production of CO2, what we generally would see is that at the ridge top, the CO2 concentrations were usually lower. And then as you got closer to the valley, the CO2 concentrations would go up for similar soil depths. And so we presume that that's because of the amount of water in the system, again, potentially enhancing some of the respiration, but also when you get to the valley, really inhibiting the diffusion rates in the system because, you know, they're just much wetter profiles when you get closer to the stream valley and the water table is so high. So like you said, in carbonate systems, this could have important implications in terms of the weathering potential of roots, depending on where you're looking in the weathering profile or in the hill slope profile. I think that answered your question.
Yeah. Okay, do we have any final questions here? All right, I don't see anything. Thanks again, Liz, for a great talk. Really interesting work, and look forward
Sure,
to
thank you for having me.
Yeah.
Thank you.
Okay, and we'll hopefully see you guys at the next talk in March.
Thanks, everyone.